Right, let's just jump straight into this explainer, because the medical stakes here are honestly mind-blowing. It might actually shock you to realize that your survival hangs in the balance of a ridiculously narrow chemical window. If your blood pH shifts even slightly below 7.35, you enter a state of acidosis. If it creeps just a tiny bit above 7.45, you're looking at alkalosis. And both of these conditions can be rapidly life-threatening. But why? Why does such a tiny, almost microscopic decimal point change have the power to completely shut down a complex human body?
Welcome back to The Explainer. Today we're unpacking this incredible, high-stakes microscopic drama of water, protons, and that crazy delicate chemical balance keeping us all alive right now. We're going to look at the invisible forces that dictate literally everything, from the shape of your DNA to the way your blood absorbs the shock of your own metabolism.
Here is our agenda for today. 1. Water, the biological solvent. 2. How water shapes biomolecules. 3. The math behind pH. And 4. Weak acids and strong buffers. All right, part 1. Water, the biological solvent.
Let's start with our first topic, and let's get one thing straight right away. Water is definitely not just some inert backdrop for biology. It is an aggressive, highly active participant in the chemistry of life. Notice this skewed tetrahedral structure? The highly electronegative oxygen pulls electrons away from the hydrogens, which creates this incredibly strong dipole, meaning it's got a partially negative end and partially positive ends. Because of this, it can act as both a donor and an acceptor, forming these super-fast, transient, 1 microsecond hydrogen bonds with an average of 3.5 other water molecules at any given moment. It is just a constantly shifting, super dynamic network. Because of that strong dipole we just talked about, water boasts this exceptionally high dielectric constant of 78.5. Now, why does that actually matter to you? Well, it means water drastically weakens the force of attraction between charged particles. If you drop salt into water, the water molecules literally swarm the sodium and chloride ions, completely shielding them from each other. That high dielectric constant is exactly what makes water the ultimate biologic solvent. It's what allows it to dissolve the massive quantities of salts and charged compounds that your cells absolutely need to function.
Okay, moving on to part 2. How water shapes biomolecules. You know, water doesn't just dissolve things. It actually acts more like an invisible sculptor, literally forcing biomolecules into their functional, life-giving shapes. To really get this, contrast to strong covalent bonds, the heavy-duty glue holding a single strand of DNA together, with the weaker, non-covalent forces we see here. It's those weaker forces, like hydrogen bonds and van der Waals interactions, that allow the DNA double helix to stack securely, but still unzip perfectly the second your cells need to replicate. But here's where water really flexes its muscles: hydrophobic interactions. Water molecules desperately want to bond with each other to maintain maximum entropy. So when they encounter something that won't play along, like a nonpolar fat, water essentially bullies those amphipathic molecules. Molecules with both water-loving and water-fearing parts, like proteins and phospholipids, are forced to fold in on themselves. They bury their hydrophobic side chains on the inside, safely away from the water, and expose only their polar, water-friendly groups to the surface. Your proteins literally take their 3D shape because water forces them into it.
Let's shift gears a bit. Section 3, the math behind pH. To really see how this builds, we have to look at the mathematical reality of what happens when these water molecules break apart. Look at this chemical equation. Water molecules are constantly colliding, right? And sometimes a proton transfers from one to another, forming a hydronium ion and a hydroxide ion. Now sure, the probability of any single hydrogen existing as an ion at a given moment is incredibly tiny, about 1.8 times 10 to the negative ninth. That means for every one pair of ions, there are nearly 2 billion intact water molecules. But because water is so highly concentrated in our bodies, this statistical phenomenon happens enough to be absolutely central to biological life. But let's be real, working with numbers like 1.8 times 10 to the negative ninth is just entirely tedious. So to handle this math practically, a scientist named Sorensen introduced the concept of pH back in 1909. pH is simply defined as the negative logarithm of the hydrogen ion concentration. This brilliant little formula takes those massive, messy orders of magnitude and just compresses them into a simple, incredibly easy-to-read scale. When we calculate pH, we follow this super straightforward 3-step process. But the absolute key takeaway here is the inverse relationship created by that negative sign. A low pH value means you have a super high concentration of free protons, a highly acidic environment. And a high pH, that means a very low concentration of protons.
Check out how this works in practice. A totally messy hydrogen ion concentration, like 3.2 times 10 to the negative 4, cleanly translates into a pH of 3.5. It takes all that complex fractional chemistry and turns it into a simple number that a doctor can read on a lab chart in a split second. A real game changer.
All right, final stretch, section 4, weak acids and strong buffers. To understand the physiological safety net that keeps you from dropping into fatal acidosis, we gotta introduce a new term here, pK. Unlike strong acids, like the hydrochloric acid in your stomach, which completely blows apart into ions, weak acids only partially dissociate, and we express the strengths of a weak acid using this pK value. It represents the exact pH level where the acid is half dissociated. The general rule of thumb to remember is the lower the pKa, the stronger the weak acid. And that perfectly illustrates the Henderson-Hasselbalch equation. Now, I know, it looks like a nightmare from high school chemistry, but honestly, it's kind of beautiful. This isn't just some abstract formula. It is the absolute mathematical proof of how your body's buffering systems actually work. It relates the pH of a solution directly to the pKa of the acid and the ratio of the base to the acid. When we actually run the numbers on this, something magical happens. If a weak acid is exactly half neutralized by a base, the concentration of the acid and the base are perfectly equal. That makes the ratio 1. And the logarithm of 1 is 0, which means that entire right side of the equation literally just drops out, leaving us with the elegant reality that the pH perfectly equals the acid's pKa. Take a look at the heavy dot right in the center of this titration curve. This right here is the climax of our biological story. Usually when you add alkali, or base, to a solution, the pH skyrockets. But look at that flat section of the S-curve right around that dot. It proves that a buffer resists pH change most effectively within 1 pH unit of its pKa. In this specific zone, the buffer simply absorbs the added ions, and the pH barely moves at all. So, how does this keep you alive? Well, just by sitting there, breathing, and metabolizing, your body is essentially a nonstop acid factory, constantly producing protons and CO2. But you don't go into acidosis, and that's all thanks to these specific physiological buffers: bicarbonate, orthophosphate, and proteins. They act exactly like that flat section of the curve we just looked at. They completely absorb the chemical shock of your metabolism, keeping your blood pH firmly locked in that safe 7.35 to 7.45 window. Ultimately, human survival isn't just about the large-scale mechanics, like your heart pumping blood or your lungs taking in air. It relies entirely, and I mean entirely, on this rapid, invisible, microscopic math of buffers and water dipoles functioning flawlessly every single second of your life. It is literally a masterclass in chemical equilibrium happening inside your veins right now. It really makes you appreciate those invisible forces holding us all together, doesn't it? Hopefully, this explainer makes you want to dive even deeper into the incredible biochemistry that builds our world. Thanks so much for joining me today. Keep learning, and I'll catch you next time. But before we go, I want to leave you with one final, kind of wild question to chew on. If water's tiny molecular dipoles stopped forming hydrogen bonds for even a single microsecond, would the very geometry of your DNA instantly fall apart?